Alright, synonyms. Okay, this is synonyms. Then you have synonyms. A bit of a tongue twister there. Okay, so what are synonyms? It's the existence of several names used for the same taxon. It's subjective synonym. And it's used to speak of all the names and history of the nomenclature of a species. Alright, it describes the relationship between two such names, and the synonym section of a taxonomic discussion is the bibliographic list. Like how we all have our bibliographic list, taxonomy also has a bibliographic list, and they have been used in published references. Okay, so rather than me just talking about it, this is what you mean by synonyms. Synonyms, sorry, blech. Alright, so here we have the species name, and then this is the author, and then in 1879. So now you're looking at the history of how the name was described. Okay, so initially, as you can see, as I've mentioned before, whenever there's a bracket, it means this is the author who first wrote it, but when he first wrote it, the name was different and he has changed over time. So if you want to track the list, this is how Studder in 1879 first wrote the species. So it was written as Paractiselba. So it's not even with the space. Okay, so it's found in this author's work in this year, and it can be found on which page? 545. Then later on, another author by the name of Ridley in 1882 also described, but with this name, and it can be found on page 101. Another author by the name of Carl Green also used this name in 1898 on page 43. However, Andres later found this and actually identified it as Paractis studeri, okay, giving credit for the first author. So I believe there's a reason why he actually had to name it different, again, by morphology, etc. So it can be found in this year on this page. And then later on, Carl Green later found that the species should be in a totally different genus, but retaining the species Alba. Perhaps there are other species within Isosycoines. So that is why they actually put it as such. So Carl Green actually wrote this in 1927 on page 52. Then later on, he did another series discussing this species in 1949 on page 42. And then is it on page 42 and also 57. So again, this is actually a style of writing. Every time you want to talk about a different author, you must put this hyphen. And the type of hyphen that you put is actually different. So it's not even a hyphen, it's called an M dash. There is N dash spelled as an EN, and there's also an M dash spelled with an EM. So you can actually look it up. Okay, like this one. This one is an M dash. This actually looks like a hyphen, but it's not encouraged. Alright. So again, this is another example. In this case, it's written with the name of the species, the scientific name, the author, and where in the paper can this figure be found. Okay, so Fontaine actually, when he first described it, it was in 1889, found on this page. PI is actually a plate. So when prior to this, a lot of taxonomy papers, of course, they use it as plates because, again, if you go back to the example of the Biodiversity Heritage Library, some of the papers, they always write the description first, and towards the end will they have plates. Sometimes you still find this in field guides, meaning to say they don't insert it directly. What I'm talking about is immediately after your descriptions, you find pictures like this. So some of the older works, what they do is it's totally just description, but towards the end of your work, only can you actually find the pictures. So, but before coming to the pictures, they will put it as plates. So they'll put it as plate 100, and then you will have the figure. So imagine this as a plate. Example, this is plate 1. So they write it as plate 1, and then they might, instead of A, B, C, they'll put 1, 2. So it's plate 1, figure 1, plate 1, figure 2, plate 1, figure 3. That's what they mean. So, and then this one can be found in plate 109, figures 1 to 3, plate, I think this is supposed to be 3, figure 6 to 7, etc. So this is how it works. All right. There's a part with terms and abbreviations used in synonyms. Okay, there's synonyms and taxonomic description. There's a lot for me to cover here, and I don't particularly feel that I would like to cover, unless if you have encountered any of this in your description or in your journals that you're looking at, you can always ask me. But probably those that I want to highlight that you may have come across, or some of you are you get confused. Okay, CF is actually very common, meaning to say that in between the species, the genus and the species name, you will find CF. So CF is where the description is to be compared with or compared with, meaning to say that the species, if I were to say example, I had the word Cymatoa CF epimerica, it means to say that the species looks like epimerica, but you're not sure of the status of it. So unless you are able to compare with the actual holotype or any type of the Cymatoa epimerica and compare your species with that, can you affirm whether it's the actual species or totally different species? So this is where CF sometimes comes in. So when I was first working with some of the species, I dealt with quite a bit with CF, where they were not sure of the status. So I had to go through a list of 300 over specimens in order to come to the conclusion that this species should retain as it is. And then com-nov or combination is where you have new combinations. Okay, so sometimes remember how with this list, sometimes you will actually have species that needs to be combined with sometimes it's the genus that needs to be combined with a new genus, and it's a list of a lot of species in that genus. So sometimes that is where genera actually have to be combined. So you will see after the species name or after the genus will be com-nov. Okay. Another one, okay, if you want to describe a new genus and a new species, you would give an abbreviation like this, gen et sp. Okay, the species is pronounced as sp sometimes. Okay, or it's just a new genus, you use gen nov. And then you have new species, you write it as nov sp. Okay, or you might actually find just an n with a sp as well. So there are different ways of writing it. Okay, and then some of you might see these terms, nomen conservandum, nomen correctum, nomen incuridium, and nomen nudum. Okay, nomen nudum sometimes is a name without a destination. This has happened a lot, where sometimes a lot of the authors back in the 1800s, 1700s have just written new species names, but without the type, without the species sometimes. So that's what you mean by a new name. It's naked because it's not equipped with all of the other descriptions and the type of the species. So sometimes it's confusing. We do not know what it is. Nomen inquirendum is sometimes species that needs to be further investigated. Why this happens sometimes is because if say the material was not found, or the species was damaged, and etc., and it has a bit of complications, you might find nomen incuridium. Nomen conservandum is species that needs to be preserved, especially if it has any it's species that, what do you call this, if it's interchangeably used all the time, and we need to conserve an old name for this. Okay, so this is where nomen conservandum comes in. So I won't go into detail again, because only when you're encountering such species then will you appreciate it better. But if you have any questions, you can always ask me, or you can check this list. How are we so far? Can I move into the next topic? Can. All right. No. Can. Okay, thank you. So now we're coming to this term etymology. Okay, now etymology is a section that occurs when a species is first described. So for all of those who are describing new species, you must have this. In modern taxonomic, written taxonomic work, you must have the word, you must have this section on etymology. So it gives a derivation of how you describe this new species. So a new species, for the respective paper, again, remember I showed you an example like nov spur, but you can also write new species. You can write spur nov spur, or nov nm. So let me just check an example here. I should have one here, which is a new species. Okay, like this one, spur nov. So it's found on page 267. Okay, so I write it as, so when I'm talking about those abbreviations, you have to write it immediately after your species and genus name. Okay, and then you will have, of course, your species, and then you will come to a section of the etymology here. So you're explaining how did you come about naming into this species. Okay, so in my case, I wrote it here like, where did I write it? Etymology. This species is named after its globular shaped body. So that's how you name it. Something like that, example. Okay. So I hope you can recap. I might not go too long with this, but remember the name bearing types is again the holotype, the neotype, your syntype, and your lectotype. The paratype is usually being para, meaning it's parallel to holotype, because the information that you've gathered is similar to that of the holotype. It's just that out of the many, like say you have 5 or 10 specimens that you have, one of this will be taken as a holotype, and the rest will be the paratype of that same locality. All right, an allotype is where this is not a name-bearing type, but is usually the opposite gender of the holotype. Okay, this is what an allotype is. Topotype is when, again, you've collected specimens of the holotype and paratype, but of a different time. It's the same locality, but of a different time. This is where topotype comes in. Syntype, again, a lot of current modern taxonomy, we don't have syntype. Syntypes is basically looking back at old material where the former authors in this, late 1700s, early 1700s, late 1800s, whatever, they haven't properly designated what a holotype and paratype is. So all of this becomes a syntype. But later on, if a later author is working on a syntype, and you're able to distinguish which one is the holotype and paratype of the author's description, so the lectotype is like equivalent to that of a holotype. And if you have other specimens, the rest becomes a paralectotype. And a neotype is only really designated when a holotype or syntype or lectotype is not able to be found. So when you're writing your material examined section, okay, all of the material that you worked with, this is a way that you have to write it. I know it looks very lengthy, it looks very, sometimes it's sort of overwhelming, but this is a style of actually writing it. So if you're going to describe about your holotype, this is where the registration number is. Some would actually put it behind the sentence, okay? And then this is the field number. If there actually is a field number written, that means to say that a station number would actually have its own registration number. But this one is where it's deposited in the museum. So these two are very different registration numbers. But this is just for people's record. And then you describe a little bit about the specimen you're working with, which is an adult female. This is caught by the collector at this year, at the location, it gives us very specific location, how the location looks like near this area. And then even gives the coordinate. And then sometimes sometimes they're very detailed in giving to exactly how far it is. So then later explains about the paratype. So in this case, it explains that it's a total of 18 specimens from the northern para, and it gives the registration number. So paratypes will have their own registration number. Okay, but most of the information will be very similar to that of the holotype. So, and then pretty much everything else is quite similar, where you will have the coordinate, the location, what year it was found, who was the collector. But if you notice here, because there are different registration numbers, it's because the paratypes are then distributed differently. So sometimes it may have, within that one big geographic region, it might have a few other localities. So that's why you might actually find some of this a little bit different, but technically it shouldn't be very different from the actual location. It's meaning to say it's like you found all of this from Bidong Island, but more specifically you want to explain where you found in Bidong Island, whether you found in Pantai China, Pantai Vietnam, etc. So if I have an example here, this is actually more probably straightforward. Material examined. Here I put the symbol of the female, which means that it's ovigerous. That means it is actually in its full reproductive form. So then I have the location in Queensland. By right, actually even have the coordinate, but because this was previously found from another collector and did not write the coordinate, so I'm not able to identify. And then what is the year was found. So being a parasitic one, you should actually write the host as much as possible. So the host also will have its own authority, and then who's the collector who found it. CSIRO, of course, meaning to say that this guy was actually from CSIRO, and this is the registration number. So sometimes material examined can be very lengthy. So like in this case, I found heaps of material examined for this particular species, Ceratata imbricata. So it was so lengthy. If you notice, yeah, sometimes it can be this lengthy that I actually had to write it as a paragraph. So because I found some of this material from South Australia, from New South Wales, from Tasmania, so I even sort of did paragraphs based on a different material that I found from different museums. So again, it will have your typical how many specimens, whether it's female or male. So sometimes you'll see the symbol here, male. In this case, I gave a description for the total length for each individuals. That's why you see all these numbers. Then again, it will typically have the location, the coordinate, the year, the collectors, etc. So that is how you write material examined. And then you have a diagnosis. A diagnosis is a brief comparative description of the organism and how does it differ from close relative. Okay, so a diagnosis is like, imagine for a medical diagnosis, you have some of the particular signs, okay, that you're sick. But then another person, patient A, has a similar signs, but his symptoms are a little bit different. So how you want to diagnose is you want to have a list of readily available, okay, if the person is sick based on fever, and if the person is sick based on, say, COVID-19 infection, per se, what is the diagnosis? So that is what it is. It's readily available, okay, in terms of the immediate diagnosis. So example, if I want to do a diagnosis, say example like with the species that I'm working with, example I have about, say example for the sake for explanation, I have about 10 different species to work with. So I will actually have a section for diagnosis. But some papers, they don't have specifically written diagnosis. They might actually have the word remarks. Okay. And usually some papers, they will have remarks written at the beginning, but some written towards the end. So, okay. So this is where example for remarks, or it's also termed as diagnosis. So how can you readily identify it? From this feature number 1, feature number 2, feature number 3, feature number 4. Okay, this is how I identify the species. If I want to make a quick comparison, how is this species different from this species, I have to use at least 2 to 3 characters. It's very different from a description, where a description will be very lengthy. Lengthy is way longer. For some reason, I don't have one here. This is already coming to my description. Okay, let me give you an example. Okay. So this is considered a description. Okay. So if you notice how descriptions, you really look into, say, example, the body, the cephalon, the frontal margin, the antennula. So you're really writing from, a description basically is writing from head to toe about the person. All right. So you can actually have like perhaps even, I don't know, 100 different characters. All of that comes under a description. But when you're coming to a diagnosis, I believe you don't want to be torturing the person to explain about all of the 100 different descriptions. So out of those 100, what is the probably top 5 or top 10 most immediate diagnosis to help you identify that species and how to differentiate from other species? So this is what a diagnosis is. Again, description, as I've shown you this now, example, this contains a straightforward and detailed description of the species. Teal, chestnut, 14 hens, etc. Again, like how I showed you this now. The style varies from group to group and publication to publication. So again, every style of writing is going to be slightly different, but ultimately the way of writing is going to be, the outcome is going to be the same. So modern journals often favor the abbreviated telegraphic style, and its non-essential grammar is left out. Okay, why do you say non-essential grammar is left out? Because you won't be writing it, example, yeah. You don't, a typical way is of writing it properly is the body of the female is subtriangular and it has 2.1 times as long as greatest width, full stop. The dorsal surface of the body is actually very smooth. That is how a typical way of writing it is, taxonomic description will be. But in this case, it removes all of those grammatical styles. So if you're talking about body, everything related about body, you will write it. Body, subtriangular, comma, 2.1 times as long as you do it, comma, dorsal surface. So all of this is related just to the body. And then when it's coming to the cephalon, which is the head, okay, cephalon, subtriangular, comma. So all of the, it's not the nouns, the adjectives, I believe. Yeah. All of that is actually, it's just commas all the way. And then if you're coming, then you have a full stop when you're talking about a different subject, the frontal margin, the pleonasms. It's like saying the body, what are all the descriptions about the body? The head, what are all the descriptions about the head? The shoulders, what are all the descriptions about the shoulders? The eyes, is it absent, present, etc. So that is how the style is actually written. Okay. How are we so far? I know it's a bit lengthy, but if you have any questions related to maybe your paper, you can ask. Okay, presumably no questions. So then I go to the taxonomic discussion. So now when you're coming to this, so technically, your description, your diagnosis, your etymology, it all, and even your material examined, these are all under the subject of results. These are your results, okay? So if you were to see example in this paper, perhaps this one. If you notice, your typical paper still has the abstract. It still has introduction. It still has material and methods. But really what comes is this part. So this part is your results and discussion. So your keys, your material examined, your diagnosis, your description, it all comes under results and discussion. However, there are some people that actually write a specific section on discussion. Okay, so the discussion part talks about the changes in the placement of the species being described. So remember that I showed you an example of, like, say, the synonym or synonymy list, where you have all the different authors changing it. Okay, so obviously in your head, you as a reader might want to know, why were these changes made? What happened? So this is where you would tackle this problem. You will discuss about why the authors changed it so and so, etc. So there will be a section for explanation of a discussion of the fact. And then when you're coming to the ecology section, so we do, again, we are not ecologists, but we help particularly ecologists with some form of information with this work. So we give other ecological information such as the habitat preference, is there any community association. So example, where I mentioned, like, if you're talking about species that are parasitic, so you would obviously want to explain about the parasites, the host, I mean, because obviously understanding the biology of the parasite, you have to understand the biology of the host as well. And the host will actually have different, say, distribution. Some are perhaps a benthic host, some are planktonic hosts, so it's different in that sense. That's why you have to tackle it differently. And then some of the species have different biology. So sometimes their reproductive seasons are actually very different. It's different from seasonal, in a seasonal aspect, how it grows, in terms of the male, female, etc. So all those has to be explained here. And again, another aspect we will look into is distribution and occurrence. Sometimes if it's not too lengthy, it also comes under the ecology section. So it's a description of the new species that may just be only one locality. For a common or wide-ranging species, it may be long list of localities. If the work is regional in scope, the author may list only its occurrences in part of the world being studied. Okay, so remember how we had a bit of a stumbling block regarding locating type specimens, right? Okay, so I'll go back to this section. And this time I've already got it prepared. Yeah. So, again, using the Museum of Natural History for Paris, again, it will be quite typical for most other museum websites. So you can look through their collection, you can look through their loan requests. In this case, example, we're looking at marine invertebrates, okay? So when we're looking at marine invertebrates, again, our example was that, okay, we're interested in decapods. So by right, you would be able to look at their collection database. And this is how it's supposed to come out, okay? So they give you a little bit of a short introduction of what estimates they have. And then this is where you can type out if you have the catalog number, the type status, and again, if you want photography, you can even choose if you want photography or not. That's one. If you have no idea what the species you're looking for, but you sort of know the scientific name, you can actually type the scientific name, and you can, or if you do not even know what it is, but you know the collectors who've been going through, or the expeditions. If it's expedition, then you will have a whole list of other species that might come out under invertebrate crustaceans. But in this case, for example, I actually know what, say, the genus I want to look for, and it's Cymothoa. So if I were to open it and I type Cymothoa, this is what is supposed to come out. So this is quite interesting because it gives you a list specifically of the types, okay? So the example in this museum, they're actually holding like the type specimen. They're also holding syntypes and also paralactotypes. So interestingly, they actually have paralactotypes of Borbonica. So the lactotypes are actually placed somewhere else, unless it's written here. But usually all of the types will actually just come out immediately here. And you can also check through their list of other specimens that they have. Okay, so if you were to just example type on, click on syntypes, it gives you a little bit about usually the set of specimens. This is your registration number. It gives you the papers that you found. Okay, so sometimes this information can be quite helpful for the curator if you want to loan the specimen. Okay, and then sometimes it might actually explain to you whether or not the specimens are based in what fixative, like it's formalin or etc. Yeah. At one point, we will be looking into the International Code for Zoological Nomenclature, clature. So we will talk about that in a later topic, so don't panic about that. But this is basically what I want to share with you about how to locate type specimens. At least this is just for the Museum of Natural History. If you look to some of the more established museums, they would actually have their collections which you can access. But if it's not available, this is where you actually have to somewhat find a way to contact the curator and actually ask questions on what they have. Okay, so pretty much that's it for my lecture. Do you have any questions? All right, so we have a question from one. Doctor, for new species, is it compulsory to write the color of the specimen in alcohol? Actually, it is very helpful. It's not compulsory, but it's actually helpful because obviously, especially like, I wouldn't just say for invertebrates, most specimens in their natural state, their colors are very more vibrant. But then in alcohol, all those colors run. And sometimes those colors will actually seep through the alcohol, and that is why sometimes your alcohol actually has some funky looking colors, okay? But usually this is after rinsing, after you've cleaned it properly. But don't forget, even your alcohol will actually have runoffs of, not say runoff, but what seeps through into the alcohol is sometimes the fatty deposit and all the moisture that is coming out from the specimen. So it actually is quite, it's not compulsory again, but it is very helpful to actually put. Because if other later authors want to identify it, it will be a bit helpful. And then the other thing is, sometimes those that are in a live state and also in the alcohol will retain the color. You see? So that's why it's very different from every species perspective. So if you can help, please write. Thank you very much for the question. I really appreciate that. It's a very good question. Any other questions? The neotype, okay. Okay, so now the neotype happens basically when, let me go back to my slide. A neotype happens, when, a holotype, or the syntype and lectotype are usually not available. Okay, so sometimes a holotype is not just the specimen itself. If the author has actually written about this, and the picture is available, it can also be used as a holotype. But sometimes some of those pictures are probably not even helpful for identification. Okay, so again, it has to be damaged, or it's the readily available specimen is not easy to find, or when I remember about talking about conserving the name of the species, it is important because sometimes it's not readily available. So maybe I can give you an example of a neotype that I've worked with during my trip to South Africa. Hang on, yeah. Okay, so this is a paper that I worked with my collaborator on designating a neotype. Now, this species actually, when this guy first described the species, okay, the specimen was not uh, was not was not found again, the type specimen, but he only had this as his picture for reference. So technically, this should be the type, okay? But the problem with this issue is that a lot of the description, if you want to compare it to modern times, is not easily recognizable, because there are so many other species that are complex in the sense that they look very similar to this. So if it's just this kind of simple photography, it's not going to be very helpful. Okay, and the thing is, a lot of the species, they occur in a very complex river system in South Africa, so all the more makes it a lot more difficult. So and this author didn't particularly describe it very thoroughly for the males and the juveniles also. So this is where we had no choice but to designate a neotype, which, other than producing the description, but we actually, what we did was, this is what we did. We took a picture of the fresh state and also those in ethanol, okay, and where it was deposited, but not just for the female. We also did for the male and also for the juvenile. Because you notice how the colors are very different. And it's interesting because once the gold color sort of dissolved, the markings actually became a bit more prominent. So I realized that these markings actually can be quite useful in description for the species. So, but all of this was not available in the previous description. So neotype designation was actually very important at this stage. And again, we tried to make sure that we give credit as much as possible to the first author, where we actually went back to the region where he first located the specimen. Even that also, he didn't have a coordinate, but because we had experts there who roughly know exactly where to pinpoint the species, so that is where we went back to the location to actually find it. So this is where it was. So this is how it is. So then we had to give the description, and then of course we had morphometrics and meristic counts. Technically, this is a diagnosis of the species, and this is in a way also part of diagnosis, because we are comparing this species with other species as well. And this is how we help description. So yeah, this is how we work in terms of coloration. If it helps, if it aids with description, that is actually very helpful. And it's even more helpful when you have more specimens to work with. Thank you very much for the question, Rosana. Any other questions? Well, welcome. All right, so if you have no any extra questions, I will actually ask questions for you guys based on the papers that you have found, and based on our discussion today of the synonym list, your headings, etc. I will actually give out the question later, and then you will have to fill up the question based on your paper. So other than that, thank you very much, and I'll see you in the next class. Bye-bye.
Terima kasih, Dokter.
Sama-sama.
Thank you, Doctor.
You're most welcome.
Thank you, Doctor.